Hello, everyone, and welcome to today's live broadcast, Complete Workflow Solution from Gene to Protein. It is presented by Michael Liss, PhD, a Senior Manager in R&D at Thermo Fisher Scientific. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. Please join me in welcoming Dr. Liss. I will now turn the presentation over to him.
Thank you for the kind introduction, and hello everybody for this little webinar about our complete workflow solutions from gene to protein. So my name is Michael Liss. I'm with GeneArt since 16 years here in Germany, and I want to tell you a little bit about our service, which we eventually have expanded and going beyond simple gene optimization and gene synthesis to actually use these genes to express and purify proteins in mammalian systems for our customers. So, a slight overview of the agenda. I will introduce you and give you a small overview about what gene synthesis actually is and what then next what gene optimization is. So the design, the in silico design of sequences prior to their actual synthesis. Then I want to give you an overview of the results of a mammalian multigene expression study, which we did with optimized genes, and tell you a little bit about the mechanisms of gene optimization. How does that actually work within the cell? Then I give you an overview of the applications and the benefits of optimized genes for expression. Talk a little bit about protein expression systems at Thermo Fisher. Then, of course, go deeply into the gene to protein services that we offer, and then it's open for your questions and hopefully my answers. So what are synthetic genes? I think by now it's really a commodity. It wasn't that way when we started with GeneArt some 16 years ago. Synthetic genes are double-stranded DNA, of course, and they can have any length. We routinely synthesize over 10 kb genes in length. And they actually can have genome size, so they are several hundred kilobases in size. The design of synthetic genes is completely free, so de novo synthesized genes can be made upon any specifications that a scientist may require. And of course, certainly all genes that we produce are 100% sequence verified. Now, how does gene synthesis work? It all starts in silico. So imagine you take a gene from the jellyfish, GFP, and you want to transfer it to the fruit fly. And of course, this works because the genetic code is universal, and we all speak the same language. However, I would say that the dialect is a little bit different. And what I mean with dialect is that the synonymous codon usage in different organisms is different. And this is where we can do an adaptation in the computer and modify, in these cases here, the wobble bases marked in red to match the most frequently used codons in the fruit fly. So this is all in silico. Now we have an electronic sequence in the computer, and the next task is, of course, to turn this into a tangible molecule. That starts with oligosynthesis. We have apparently Europe's largest oligosynthesis lab here in Regensburg in Germany. but to date we entirely use it for gene synthesis. So here, of course, we have the 4 amidites, A, C, G, and T, and on the machines they are made into oligos which have a length something between 50 or 100 nucleotides. They are designed in a way that they overlap each other, like you will see on the next picture here. The animation starts. Sorry. Apparently it does not start with the animation, but anyway, so they're designed in a way that the oligos, they overlap each other to eventually assemble a full-length molecule. That molecule then is not covalently linked, but these missing gaps of the oligonucleotides are filled in with polymerases, and after a few cycles of cycling, basically this primer extension reaction leads to full-length molecules, which can then be amplified. Sorry, now you will see the animation. So the oligos, they find each other, the gaps are filled in with polymerases, and you end up with a full-length molecule, which can then be amplified and cloned into a suitable vector, transformed into E. coli, and colonies can be picked to verify the correct sequence of a monoclonal molecule. That's basically how gene synthesis works, and the overall process, depending on the workflow that you use, may take about 5 days. As I said before, gene synthesis has become a true commodity over the years, and to date we have many customers that don't really bother cloning anymore and amplifying a sequence from some template in the fridge, using primers to modify restriction sites and so on, but they just order the sequence that they have in the computer anyway. Synthetic genes are today used in all kinds, in all areas of biotechnology, like basic research. They're used in functional assays, for antibody production, for protein production, actually also for optimization of proteins and enzymes if you adapt synthetic genes to directed evolution, which is possible. You can use synthetic genes as positive controls for DNA detection assays. You can use them for cell line optimization, actually for anything you can think of where you need DNA. So in this picture, this is kind of the portfolio that we currently offer within synthetic biology at Thermo Fisher GeneArt. Of course, we help our customers to design projects according to their specific needs. We do gene optimization and gene synthesis, which I just explained a little bit. And we can make gene variants. If you, for example, want to do an alanine scan and require several hundred or of different similar sequences, we can do this as well. We can make gene libraries for phage display, for example, creating huge diversity in these synthetic genes. Subcloning is actually a big part of our portfolio, because we have many customers who say, yes, I want a synthetic gene, but please provide it in this and that vector. Then plasmid production up to milligram levels, and then also cell lines and proteins, which I'm going to focus on now. I want to explain a little bit what gene optimization is and how it works. So, as I mentioned before, biotechnology is possible, or is actually easy, because the language of all the organisms is basically the same, and it's easy to transfer a gene from one organism to another and have a good chance that it actually works. However, here's a small part of some codon usage tables of 4 organisms: a plant, human, yeast, and E. coli. And if you look at the 6 synonymous arginine codons and look at the columns of humans and E. coli, you will notice that humans with the blue shaded numbers prefer some different codons than E. coli, the blue shaded codons. And vice versa, the rare codons in E. coli are the most frequently used codons in Homo sapiens, and so on. That means if you, for example, take an insulin gene from a human and plant it into bacteria, it will work. However, it will work with not the most frequent codons, and that may have an impact on the expression levels of that specific insulin protein. That's about codon usage. Codon usage is not the only parameter in the dialect of the different organisms, I would say. There are other parameters that can be adapted as well. And that's, for example, the GC content. Everybody knows that different organisms have a different GC content, and it's a good idea to adapt the GC content to the host in order to facilitate or to guarantee genetic stability, good performance, and also good expression. Then, for example, cryptic splice sites may be an issue. If you go from, if you express in a mammalian cell, it may be a cDNA or a gene from another species, then it may happen that there's a cryptic splice site and the mRNA is not processed correctly, and you end up with the zero expression. There's actually a very famous example when they first tried to express jellyfish GFP in tobacco. They got zero expression, and they found that the jellyfish cDNA contains a cryptic splice site which is recognized by the plant, and the DNA, the RNA is not processed correctly. Premature polyadenylation signals may also be an issue in faulty mRNA design. Direct repeats in the DNA may lead to instability, genetic instability. Indirect repeats may lead to RNA secondary structure, which also hamper translation. And then, for example, in plants, there's so-called killer sequences. That's AT-rich sequence or sequence structures that have a impact on the mRNA half-life and lead to a faster degradation of the mRNA. So all these parameters can be taken into account when you optimize gene in silico. Now, optimization of a gene in silico is actually not that trivial, and that is because the number of sequences coding for one and the same protein is astronomically high. It's the number of combinations of all the synonymous codons. So how did we solve that problem? I show you here a short animation of how our algorithm basically works. So you see that our algorithm for this short peptide would come up with the most preferred codon for almost all amino acids, but the threonine is coded by the second most frequent codon. For what reason? In this specific case, I don't know. Maybe the algorithm said, okay, I rather use ACA instead of ACC, because ACC would push the GC content over a certain threshold. Or splice site would be generated, or maybe a restriction site would be generated, which should be included, and so on. So I mentioned that the number of combinations of different codons is astronomically, and how does our algorithm solve that? It's a sliding window approach. So imagine that in the first row you see a small sliding window covering 6 different codons, And for these 6 codon positions, it generates all possible combinations and chooses the best one fitting to all the parameters, GC content, most frequent used codon, no splice sites, and so on. Then the first codon is fixed. The sliding window moves one amino acid further, and it does the same thing again. Then the first codon is fixed, and so on. And that way it slides over the complete window, the complete open reading frame. That's our approach, and it leads to a sequence which uses the most frequent codon as much as possible, but it also takes all the other parameters into account. Now about protein production. Next thing. Many scientists, when they think about protein production, think about E. coli, because E. coli is fast, it's easy handling, it's cheap, it's well studied, but expression in E. coli has a number of disadvantages. It has no post-translational modifications, no membrane environment for membrane proteins. It often leads to misfolding and aggregation and inclusion bodies. It has some restrictions to large size or oligomeric proteins, and of course no post-translation, so also no glycoproteins are possible. Mammalia, of course, circumvents all these disadvantages of E. coli, but has its very own obstacles. Sometimes mammalian expression has some transcriptional silencing. The mRNA may be unstable in the cell. Alternative splicing may happen, premature polyallelation, or in general the translation is inefficient. And all these things, they can be overcome through a good design of the gene before you synthesize it. We have actually tested this in a study a couple of years ago, which we have published, and you will find the reference in a few slides. We have taken 50 genes, 50 human genes, and have synthesized them as a wild-type version, and have optimized them and synthesized them in a optimized version. You may ask, okay, why optimize human genes for human expression? Because we then took these into human cells and expressed them. But of course, our genes in our body are not optimized for highest yield, but they are optimized for highest performance in the overall context of the cell. So expression, maximum expression is of course never a goal of or almost never a goal of the evolution and the design of our genes. So here's some snapshots of the outcome. All the transfections and quantifications on a Western blot were done in triplicates, and you see for CREB1 a transcription factor in the top row that it's noticeable on the Western blot very nicely that the optimized version had a higher expression than the wild type, and in that case it's a 2.8-fold increase of expression. And then the same is true for the other ribosomal protein, for protein kinases, for membrane proteins, for cytokines. So on the top row, IL-2, we actually did not detect any expression when we transfected the wild-type cDNA, but with the optimized, we got a faint band. So in that case, it's from 0 to something expression. And overall, for the 50 genes, we observed that in 96% of the cases for these 50 proteins, we got an equal or a higher protein expression. Actually, in 86%, the optimized version gave a significantly higher expression than the wild type. In 10%, it was equal, and only in 2 cases, so 4%, the optimized gene expressed less than the wild type. So what that means is overall you get a higher expression, but also the reliability that you get a detectable expression at all is much, much higher. Overall, we got a 25-fold increase, up to 25-fold increase on protein yields, and only 2 of those cases were actually did perform less than the wild-type version. So all these tests were made in 293 cells, and we wanted to see whether it we get the similar results in Chinese hamster cells and in SF9 insect cells. And basically, you see that with the green arrows, the results were very comparable. So gene optimization worked equally well in 293 and in CHO cells. And in one case, in the insect cells, it was in the HEC293 cells, it was less. And even in the SF9 cells, the expression rate was higher as compared to the wild-type versions. Now, what's the mechanism behind gene optimization? I explained the parameters, codon frequency, GC content, but what's the what's the concepts behind optimization, and why does it lead to a higher expression? We have tested, in this case for MIP1 alpha, several parameters in the transfected cells. So you see, obviously it has an effect on the overall expression level of the protein. In that case, with the optimized version, we got a 3-fold better expression as compared to the wild type. If we looked at the de novo synthesis rate, so the nuclear run-on rate of transcription, we also found a 30% increase of that particular step in the overall expression or transcription and translation context. Also, if we looked at the mRNA stability, we found a 14% increase of the stability of mRNA, probably due to a slightly higher GC content as compared to the wild type. Then on a northern blot, we found that the optimized mRNA did not perform any better when it comes to nuclear export. So wild type and optimized mRNA were equally well exported from the nucleus into the cytoplasm. And the mRNA steady state levels were 18% higher. That's probably an effect that comes from both, from the de novo synthesis rate, so the nuclear run-on of the mRNA, and the mRNA stability in combination gave 80% higher mRNA abundance inside the cell. And overall, of course, the translational efficiency was 20% higher, so that means that the mRNA, once it is there, the efficiency of turning this into a protein on the ribosome was also 20% higher as compared to the wild type, maybe due to the lack of any or the elimination of secondary structures in the mRNA. When we optimize genes for our customers, we often get the question, yeah, okay, I see that the expression rate can be increased, but will the final protein be functional? I tend to answer that this is a general question in biotechnology. Whenever you have a gene from one organism to transferred from one organism into another, then you are in a, I would say, codon context that is not natural in terms of it is adapted by evolution. But in this case, So that's the same thing whether it's from another organism coming into a cell or whether it's synthetic coming into a cell. It's not. There's no conceptual difference. However, of course, you can argue if a protein is expressed really high, then maybe it lacks the time for proper folding and may lead to a larger proportion of non-functional protein. So I will show you here an example of 3 proteins, kinases in that case, which we expressed in also again coming from wild type DNA or from optimized cDNA. And for and they all expressed better from the optimized genes. And for junk kinase 1, we did a phosphorylation assay. And in a nutshell, what we found is that the protein coming from the wild type gene, of course, was able to give a positive kinase signal, but this junk kinase coming from an optimized gene had the very same performance, which you see on the very right side with a phosphate-specific antibody. So there was no difference. So in that case, we could show that both proteins, or the same protein coming from both genes, had a very comparable function. Another interesting thing that you can do with optimized genes. Now, the optimized gene has a different sequence than the wild type gene. That means that the optimized genes will react differently to an siRNA than the wild type gene. So if you design a siRNA against the wild type gene, it's unlikely that, or you can design the optimized gene in a way that the siRNA will not silence the optimized gene. And we did this with cyclin-dependent kinase 2, which is a cell cycle regulator. And you see on the in the FACS plot on the top left, this is the untransfected cell. As usual, most cells are in the G1 phase, and only few cells are in the G2 phase. If we transfect an siRNA against the wild type CDC2, then more cells are arrested in G2. Now, if we transfect this siRNA together with an optimized gene for CDC2, then CDC2 can be expressed again, and this siRNA cannot silence the optimized version. And on the bottom left, you see that again the fraction of cells arrested in G2 decrease. So that's a very interesting function to or feature of optimized genes to validate siRNA functionality. So the summary of the study is that we could show with 50 genes that gene synthesis and gene optimization really increases the overall expression and the success rate of successful protein expression. It improves general yields, and it does not alter the solubility or the activity of encoded protein, and allows to confirm siRNA phenotypes. Now, there are, of course, other companies out there, competitors, who also offer gene synthesis and gene optimization. And we have taken a closer look about different optimization strategies, so in silico optimization strategies. Some of them are very different from ours, and some of them are similar to ours, I would say. We have synthesized such genes from different competitors, designed from different competitors, and tested them in a mammalian expression analysis here. And so in a nutshell, what you see here is that, again, the GeneArt optimized genes had a about threefold higher expression than the wild-type gene. But then there's also expression levels from genes optimized from competitors, and they are all expressed less than the GeneArt, not just the GeneArt designed gene, but even less than the wild-type gene, which of course is not very favorable because you typically want to increase and not decrease your expression. So on this slide, what I just want to focus you on is that the protein service that we offer is not the only service that we have in our portfolio around gene synthesis. There's many other products which most of you are likely to be familiar, but here in this particular talk should be focused on the proteins and the expression in HEK, in CHO, and in insect cells. For the expression, we of course rely on the excellent reagents that Thermo Fisher allows us to use, and that is of course freestyle, and since a year for CHO, or since 3 years for 293 cells, the XP system. And what does a service look like? There's different kinds of requests from customers, and we have shaped our services around these different needs from customers. So if you would need a protein to be synthesized, we need from you an amino acid sequence electronically or an accession number, and that's basically it. But then you also need to specify on your expectations, so the yield that you need. This can be a specified culture volume. You may say, okay, please deliver whatever you get from 1 liter culture. And you can, of course, also specify which cells you prefer, whether it's 293 or CHO cells or insect cells. And in that case, you would, of course, get the synthesized gene in an expression plasmid. You would get the purified protein from that specified culture volume, or you may also get the rare raw culture supernatant or the cell pellet. If you need a specified protein amount, then that's also possible, also with the same cells and with the same deliverables, but we need to do a gene-to-protein pilot first. So what we would do then is we make a small culture volume, transfect that expression construct, do the expression, also do a time course about the harvest day, do a purification, and then we know what yield we got from that particular culture volume, and can then upscale accordingly to meet the specified protein amount that you requested. So next I want to talk a little bit about some customer success stories, show you some examples on how gene optimization and gene synthesis, and in conjunction with the XP systems, works or gives better performance for our customers. This first example is still without the XP system. In that case, we made all 9 combinations of 3 different light chains with 3 different heavy chains for a customer. And the customer already gave us some, on the top left, table, you see gave us some expression yields that he was able to achieve. And on the right hand table, you see what expression levels we achieved after we optimized the genes and expressed that. And down there, you see the same results also in a bar. So the overall increase was about 20 to 50-fold, and in one case, even 650-fold increase. The next example was different iterations, I would say, of combinations of gene, vector, and production cell line. So six different antibodies from a customer, and the white bars in the graph represent the wild-type gene and customer in-house production, those expression levels, and then the dark blue bars are already optimized gene, but in the customer vector and with the freestyle 293 cells, so already an increase. Then the gray bars is optimized gene, but now in pcDNA 3.3 vector and in a freestyle system. In some cases for antibody 3 and 4, that gave a nice increase. And then the bright blue bars are then everything optimized, so optimized gene, pcDNA 3.3 and XP293, which in almost all cases gave a much higher yield of protein expression. For the sake of time, I will touch this only very loosely because I have some others. So here's some antibody expression results. Comparison of XP293 versus XP-CHO, and it basically shows that both expression systems work remarkably good and give a well-folded antibody. You see that in the chromatogram HPLC plot down there, and heavy and light chain are well folded and into a full functional antibody. This is what a documentation of a protein looks like. It's a little bit hard to read, I think, on a small screen. However, what I want to say here is that you get or our customers get full documentation about all the aspects that were used from gene synthesis to protein production and purification. So starting from the sequence, of course, is documented, then which vector is used, with which expression cells, the culture volume, how it has been purified, what the endotox levels are of the protein, the concentration, how it looks on a Comasi gel, what the HPLC purification looked like, which fractions have been pooled for the protein, and with a final chromatogram of the then purified protein. This documentation is highly customizable and is usually discussed in advance with the customer on each and every project. Here I can show you how easy it is to order proteins, synthetic proteins. So on the Thermo website, actually it's easiest to just Google gene synthesis Thermo Fisher. You find a GeneArt Gene Synthesis tab, and if you click on that, there's a link to GeneArt cell lines and proteins. And on the website that you reach then, there's a small download button that allows you to download a request form, which you can then email to the given address. That's basically it. And then on that form, you can enter, of course, the sequence or the accession number and what purification method you need, what yield you need, and so on. So already going to the overview, what I hope I was able to show you is that with our gene optimization, we can increase significantly the protein yield in the mammalian system, of course also in other systems, but here we're focusing on mammalian expression. There's full design flexibility if you want to use the gene further and require specific restriction sites or Gibson cloning design for Gibson cloning, then this can all be included in the design. We, of course, use the in vitro gen vector suite that's compatible with the XP system. We have a high-quality plasmid preparation in order to basically to minimize endotox levels in the protein. And the whole process is done in-house, and that, of course, allows for best turnaround time from the electronic sequence to the purified protein. And I don't want to read that out loud. We express in 293 cells in XP and freestyle system, and also in CHO cells, and in back-to-back SF9, SF21, and high-5 systems. We of course, purify the proteins on FPLC and HPLC on different columns, which are, of course, project-specific and upon customer needs. We analyze the proteins that we synthesized with standard page, 280 nanometer quantification, also with a blitz if it is an antibody with HPLC, and we are just setting up a mass spec then, so that will be part of our analysis QC as well. So the gene to protein, I said in record time, the overall gene to protein service typically takes 30 business days, but can be faster in times. That really depends on specific project, like the size of the gene and the required yields of the protein and so on. The performance is extremely reliable, so our gene synthesis reliability is about 100%, and then it comes to proteins. There the results are a little bit more varying, but we usually almost always get measurable expression, but of course that's very much protein dependent. The protein expression is extremely scalable and very flexible, so we produce from 100 milliliter cell culture volumes up to 25 liter wave-back volumes. And the whole system is very secure. Of course, the whole e-commerce system is ISO certified, and of course our staff is trained also accordingly. And with that, I hope that I have given you some overview of our gene-to-protein services at Thermo Fisher GeneArt, and I'm happy to take some of your questions if you may have some. Thank you.
Thank you, Dr. Liss. Thank you, Dr. Liss, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you recommend incorporating signal sequences in the construct to express membrane proteins?
If it's a membrane protein, then of course it already has a signal sequence, if it's a mammalian membrane protein, that is. We do an analysis of the sequences prior to the design. If we detect a single sequence, then we know it's secreted or ends up in the membrane. If it does not have one, then we discuss with the customer whether you want it to have it secreted. Then we can add an artificial signal sequence in order to secrete it. Or if it comes from another organism and should end up in the membrane, to end up in the membrane. Yeah.
Thank you. What is the major factor at the level of transcription for a non-expressor or poorly expressed protein?
Can you repeat that question, please?
Yes. What is the major factor at the level of transcription for a non-expressor or a poorly expressed protein?
I mean, that's a hypothesis that it's if it's poorly expressed, that it is due to low transcription. I would say my best answer is, of course, it's a matter of a good promoter in order to have a high, a good transcription. Second is that the 5' end of the mRNA should be somehow relaxed, maybe a little bit, be a little bit more AT rich, in order to facilitate transcription. But then I think what has more impact on the level of expression in terms of mRNA or RNA is not so much the transcriptional activity, but more the stability of the mRNA in the nucleus. So if you think about cytokines, for example, of course they're expressed extremely well upon inflammatory signal, for example, in our cells. But they should be expressed only for a short time, and therefore they have a very short half-life. The mRNA have a very short half-life, so that this protein concentration diminishes quickly enough. And the cytokines make this possible due to specific AT-rich regions in the mRNA.
Thank you. Apart from codon optimization, does Kozak sequence have any major effect?
We typically include a Kozak sequence. I personally, and that's a religious question. I personally don't believe, or I haven't seen much convincing data that the Kozak sequence really helps. We include it because still it appears quite a bit in the literature that it's state of the art to include a Kozak sequence. And for us, it's easy. Yeah, it's a few nucleotides more, and doesn't add to the cost, and it doesn't harm, at least. So we typically add a Kozak sequence, but we haven't seen convincing evidence that it actually helps.
The next attendee says, thank you, and asks, is the optimization free or connected with an order?
Again, can you repeat that, please?
Yes. Is the optimization free or connected with an order?
Free.
Great.
Simple as that, yeah.
Great. How long does it take to receive a protein, and what is your turnaround time?
Yeah, that depends a little bit on the overall scope of the order. So for the most comprehensive service, you send us a sequence electronically. We synthesize the gene, do a larger scale plasmid preparation, transfect a liter of cells, and incubate the cells for 6-7 days, then harvest, purify, polish the protein, do the fill and finish and the quantification. That takes about 30 business days. It can be faster if the scale is lower, or sometimes customers say, I have the expression plasmid already, I'm going to send you that. So we can skip the gene design and the synthesis, gene synthesis part, but can start right away with plasmid preparation and with transfection. Then it's shorter. But I think around about 30 business days is a very good number to start with.
This next one is a two-parter. I'll read both parts in case it's helpful, and then let me know if you need me to repeat the second part. Do you also codon optimize the leader sequence? And is it sometimes better if the translation in the leader holds for allowing a better folding?
Yes, we do optimize the leader sequence, and we have no observation that the optimization hampers in any way folding or functionality of the protein. I would say that 80% of the proteins that we are making are actually antibodies, and of course here the demand is that the expression rates are as high as possible in the yields, and so we use we have never seen that antibody lacks folding or proper phenotype upon optimization of the leader peptide.
Okay, thank you. The next one is, when should somebody prefer insect cell expression over mammalian cell expression?
That's a good question. I think I can't give you a good answer to that because I personally haven't worked with insect cells before. From what I know, though, is that some proteins express extremely well in insect cells. And of course, the nice thing is that if you choose the insect expression system, then the first thing that you do is you generate virus, baculovirus, and with that you infect the cell. So that is kind of a perpetuative source of reinfection of new cells. So it's once you have the virus, it's very easy and very fast to make new protein batches. This I see as one of the advantages, and then from what I've heard is that some proteins just express extremely well in insect cells.
Do you incorporate 5' UTR for protein production purposes?
Apart from the Kozak sequence, usually not. There are within the 5' UTR are within the vector, and the PC3.3 that is preferred for the XP system already contains all these parts, which is essentially a really strong promoter. And other than that, nothing else is necessary, no.
Which purification methods do you offer, including epitope tags?
So routinely, as I said, we 80% of the proteins that we're doing are antibodies. So of course these we typically purify with the FC, with their FC part, with protein A, and gel filtration for polishing. But we also use other tags like his tag, strep tag, flag tag, or other customer specified tags. We can also cleave the tags upon expression. Here we usually use TEF, but can also use other proteases for cleavage of the tags.
Do you offer membrane protein purification?
No. Membrane purification is a very specific, I would say, art already, and it can become very tedious and very protein specific. We do offer membrane protein expression, but in these cases we typically deliver only the cell pellet. But of course we verify the expression with a Western blot, but then leave it up to the customer to use the cell pellet for further use of the membrane protein.
What is part of your QC, or what is included in the documentation?
I have shown a documentation before. So as for the protein, it's a standard Comassi gel of denatured and native protein. Then the concentration as measured by OD280, Western blot. Then for antibodies, we usually do a protein A-based blitz titer determination. And then optional are other QC methods like HPLC, or an endotox test is also optional, or mass spec, things like that. Mm-hmm.
How much protein will I get if I order your service?
It's highly dependent on the protein itself, of course. So if you order the volume option, which is please transfect a liter of culture and get me whatever you can express from that, then it's open what the expression levels will be. So from antibodies, I can tell you that typically from a liter of transfected cells, we achieve 100 to 300 milligram of antibody. But it also depends on the antibody itself. But if you say, no, I want 100 milligram of this and that protein, then we have to do a pilot first, and then a second scale up on volume according to the results of that pilot.
Okay, we have time for one more question. I am surprised to see your results that codon optimization from different vendors work very differently. Major differences.
That's not really a question.
Yeah, I think it's easy.
But
comment?
I can understand that surprise, and I would. This is kind of a showcase, but we have done these studies for other proteins as well, and we see maybe not that extreme, but we see very similar results. So you can make that experiment on your own. You can go to different vendors and do the optimization on their website and see the results. You can hit the optimize button several times, and from some vendors with their algorithm, you will see that every time the sequence will be different when you optimize it. So it's a stochastic algorithm. That's one thing that would make me suspicious. What you can also do, you can take a sample sequence, optimize it at different vendors, and then look at the GC content. And you will see differences in the GC content. And then if you look at what is the optimal GC content for mammalian expression, just by looking at the sequence or the GC content, you will observe that some of them may have a higher likelihood to express better than others. So this is an experiment I would suggest you to if you are interested, to dig into this to get more confidence in this. But I really can assure you that the results that I have shown you may be extreme, but are kind of representative from what we observe with many different other proteins as well.
Well, I would like to once again thank Dr. Liss for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2019. Please share with your colleagues who may have missed today's live event. Also, stay tuned for our next presentation, Cryo-Electron Microscopy, Revolutionizing the World of Structural Biology and Healthcare. It is presented by Mark M.H. Storms, a Product Marketing Manager in the Life Sciences Business Unit of the Materials and Structural Analysis and Analytical Instruments Group at Thermo Fisher Scientific. You won't want to miss it. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Thank you. Goodbye, everybody.